The enzyme-linked immunosorbent assay is a test that uses antibodies and color change to identify a substance. ɨʼlaɪzə is a popular format of wet lab-type analytic biochemistry assay that uses a solid-phase enzyme immunoassay to detect the presence of a substance, usually an antigen, in a liquid sample or wet sample. The ɨʼlaɪzə has been used as a diagnostic tool in medicine and plant pathology, as well as a quality control check in various industries. Antigens from the sample are attached to a surface. Then, a further specific antibody is applied over the surface so it can bind to the antigen. This antibody is linked to an enzyme, and, in the final step, a substance containing the enzyme's substrate is added. The subsequent reaction produces a detectable signal, most commonly a color change in the substrate. Performing an ɨʼlaɪzə involves at least one antibody with specificity for a particular antigen. The sample with an unknown amount of antigen is immobilized on a solid support either non-specifically or specifically. After the antigen is immobilized, the detection antibody is added, forming a complex with the antigen. The detection antibody can be covalently linked to an enzyme, or can itself be detected by a secondary antibody that is linked to an enzyme through bioconjugation. Between each step, the plate is typically washed with a mild detergent solution to remove any proteins or antibodies that are specifically bound. After the final wash step, the plate is developed by adding an enzymatic substrate to produce a visible signal, which indicates the quantity of antigen in the sample. Of note, ɨʼlaɪzə can perform other forms of ligand-binding assays instead of strictly immunoassays, though the name carried the original immuno, because of the common use and history of development of this method. The technique essentially requires any ligating reagent that can be immobilized on the solid phase along with a detection reagent that will bind specifically and use an enzyme to generate a signal that can be properly quantified. In between the washes, only the ligand and its specific binding counterparts remain specifically bound or immunosorbed by antigen-antibody interactions to the solid phase, while the non-specific or unbound components are washed away. Unlike other spectrophotometric wet lab assay formats where the same reaction well can be reused after washing, the ɨʼlaɪzə plates have the reaction products immunosorbed on the solid phase which is part of the plate, so are not easily reusable. Principle: as an analytic biochemistry assay, ɨʼlaɪzə involves detection of an analyte in a liquid sample by a method that continues to use liquid reagents during the analysis that stays liquid and remains inside a reaction chamber or well needed to keep the reactants contained. It is opposed to dry lab that can use dry strips, and even if the sample is liquid, the final detection step in dry analysis involves reading of a dried strip by methods such as reflectometry and does not need a reaction containment chamber to prevent spillover or mixing between samples. As a heterogeneous assay, ɨʼlaɪzə separates some component of the analytical reaction mixture by adsorbing certain components onto a solid phase which is physically immobilized. In ɨʼlaɪzə, a liquid sample is added onto a stationary solid phase with special binding properties and is followed by multiple liquid reagents that are sequentially added, incubated and washed, followed by some optical change in the final liquid in the well from which the quantity of the analyte is measured. The qualitative reading usually based on detection of intensity of transmitted light by spectrophotometry, which involves quantitation of transmission of some specific wavelength of light through the liquid. The sensitivity of detection depends on amplification of the signal during the analytic reactions. Since enzyme reactions are very well-known amplification processes, the signal is generated by enzymes which are linked to the detection reagents in fixed proportions to allow accurate quantification, thus the name enzyme-linked. The analyte is also called the ligand because it will specifically bind or ligate to a detection reagent. Thus ɨʼlaɪzə falls under the bigger category of ligand-binding assays. The ligand-specific binding reagent is immobilized, that is, usually coated and dried onto the transparent bottom and sometimes also side wall of a well, which is usually constructed as a multiple well plate known as the ɨʼlaɪzə plate. Conventionally, like other forms of immunoassays, the specificity of antigen-antibody type reaction is used because it is easy to raise an antibody specifically against an antigen and bulk as a reagent. Alternatively, if the analyte itself is an antibody, its target antigen can be used as the binding reagent. History: before the development of the ɨʼlaɪzə, the only option for conducting an immunoassay was radioimmunoassay, a technique using radioactively labeled antigens or antibodies. In radioimmunoassay, the radioactivity provides the signal, which indicates whether a specific antigen or antibody is present in the sample. Radioimmunoassay was first described in a scientific paper by Rosalind Sussman-Yellow and Solomon Berson published in 1960. Because radioactivity poses a potential health threat, a safer alternative was sought. A suitable alternative to radioimmunoassay would substitute a non-radioactive signal in place of the radioactive signal. When enzymes react with appropriate substrates, a change in color occurs, which is used as a signal. However, the signal has to be associated with the presence of antibody or antigen, which is why the enzyme has to be linked to an appropriate antibody. This linking process was independently developed by Stratis Avramias and Gia Pierce. Since it is necessary to remove any unbound antibody or antigen by washing, the antibody or antigen has to be fixed to the surface of the container. That is, the immunosorbent must be prepared. A technique to accomplish this was published by Wide and Jerkaporath in 1966. In 1971, Peter Perlman and Eva Envall at Stockholm University in Sweden, and Anton Skous and Bork van Weemen in the Netherlands independently published papers that synthesized this knowledge into methods to perform ɨʼlaɪzə. Traditional ɨʼlaɪzə typically involves chromogenic reporters and substrates that produce some kind of observable color change to indicate the presence of antigen or analyte. New ɨʼlaɪzə-like techniques use fluorogenic, electrochemiluminescent, and quantitative PCR reporters to create quantifiable signals. These new reporters can have various advantages, including higher sensitivities and multiplexing. In technical terms, newer assays of this type are not strictly ɨʼlaɪzəs, as they are not enzyme-linked, but are instead linked to some non-enzymatic reporter. However, given that the general principles in these assays are largely similar, they are often grouped in the same category as ɨʼlaɪzəs. In 2012 an ultrasensitive, enzyme-based ɨʼlaɪzə test using nanoparticles as a chromogenic reporter was able to give a naked-eye color signal from the detection of mere attograms of analyte. A blue color appears for positive results and red color for negative. Note that this detection only can confirm the presence or the absence of analyte, not the actual concentration. Types: Indirect ɨʼlaɪzə. The steps of indirect ɨʼlaɪzə follows the mechanism below. A buffered solution of the antigen to be tested for is added to each well of a microtiter plate, where it is given time to adhere to the plastic through charge interactions. A solution of known reacting protein, such as bovine serum albumin or casein, is added to any plastic surface in the well that remains uncoated by the antigen. The primary antibody is added, which binds specifically to the test antigen coating the well. This primary antibody could also be in the serum of a donor to be tested for reactivity towards the antigen. A secondary antibody is added, which will bind the primary antibody. This secondary antibody often has an enzyme attached to it, which has a negligible effect on the binding properties of the antibody. In other cases, as in the diagram to the left, the primary antibody itself is conjugated to the enzyme. A substrate for this enzyme is then added. Often, this substrate changes color upon reaction with the enzyme. The color change shows the secondary antibody has bound to primary antibody, which strongly implies the donor has had an immune reaction to the test antigen. This can be helpful in a clinical setting, and in research. The higher the concentration of the primary antibody present in the serum, the stronger the color change. Often, a spectrometer is used to give quantitative values for color strength. The enzyme acts as an amplifier. Even if only few enzyme-linked antibodies remain bound, the enzyme molecules will produce many signal molecules. Within common sense limitations, the enzyme can go on producing color indefinitely, but the more primary antibody is present in the donor serum, the more secondary antibody plus enzyme will bind, and the faster the color will develop. A major disadvantage of the indirect ɨʼlaɪzə is the method of antigen immobilization is not specific. When serum is used as the source of test antigen, all proteins in the sample may stick to the microtiter plate well, so small concentrations of analyte in serum must compete with other serum proteins when binding to the well surface. The sandwich or direct ɨʼlaɪzə provides a solution to this problem, by using a capture antibody specific for the test antigen to pull it out of the serum's molecular mixture. ɨʼlaɪzə may be run in a qualitative or quantitative format. Qualitative results provide a simple positive or negative result for a sample. The cutoff between positive and negative is determined by the analyst and may be statistical. 2 or 3 times the standard deviation is often used to distinguish positive from negative samples. In quantitative ɨʼlaɪzə, the optical density of the sample is compared to a standard curve, which is typically a serial dilution of a known concentration solution of the target molecule. For example, if a test sample returns an OD of 1.0, the points on the standard curve that gave OD equals 1.0 must be of the same analyte concentration as the sample. Sandwich ɨʼlaɪzə. A sandwich ɨʼlaɪzə is used to detect sample antigen. The steps are: a surface is prepared to which a known quantity of capture antibody is bound. Any non-specific binding sites on the surface are blocked. The antigen-containing sample is applied to the plate. The plate is washed to remove unbound antigen. A specific antibody is added, and binds to antigen. Enzyme-linked secondary antibodies are applied as detection antibodies that also bind specifically to the antibody's Fc region. The plate is washed to remove the unbound antibody enzyme conjugates. A chemical is added to be converted by the enzyme into a color or fluorescent or electrochemical signal. The absorbency or fluorescence or electrochemical signal of the plate wells is measured to determine the presence and quantity of antigen. The image to the right includes the use of a secondary antibody conjugated to an enzyme, though, in the technical sense, this is not necessary if the primary antibody is conjugated to an enzyme. However, use of a secondary antibody conjugate avoids the expensive process of creating enzyme-linked antibodies for every antigen one might want to detect. By using an enzyme-linked antibody that binds the Fc region of other antibodies, this same enzyme-linked antibody can be used in a variety of situations. Without the first layer of capture antibody, any proteins in the sample may competitively adsorb to the plate surface, lowering the quantity of antigen immobilized. Use of the purified specific antibody to attach the antigen to the plastic eliminates a need to purify the antigen from complicated mixtures before the measurement, simplifying the assay, and increasing the specificity and the sensitivity of the assay. A descriptive animation of the application of sandwich ɨʼlaɪzə to home pregnancy testing can be found here. Competitive ɨʼlaɪzə. A third use of ɨʼlaɪzə is through competitive binding. The steps for this ɨʼlaɪzə are somewhat different from the first 2 examples. An labeled antibody is incubated in the presence of its antigen. These bound antibody-antigen complexes are then added to an antigen-coated well. The plate is washed, so unbound antibody is removed. The secondary antibody, specific to the primary antibody, is added. This second antibody is coupled to the enzyme. A substrate is added, and remaining enzymes elicit a chromogenic or fluorescent signal. The reaction is stopped to prevent eventual saturation of the signal. Some competitive ɨʼlaɪzə kits include enzyme-linked antigen rather than enzyme-linked antibody. The labeled antigen competes for primary antibody binding sites with the sample antigen. The less antigen in the sample, the more labeled antigen is retained in the well and the stronger the signal. Commonly, the antigen is not first positioned in the well. For the detection of HIV antibodies, the wells of microtiter plate are coated with the HIV antigen. 2 specific antibodies are used, one conjugated with enzyme and the other present in serum. Cumulative competition occurs between the 2 antibodies for the same antigen, causing a stronger signal to be seen. Sera to be tested are added to these wells and incubated at 37 degrees Celsius, and then washed. If antibodies are present, the antigen-antibody reaction occurs. no antigen is left for the enzyme labeled specific HIV antibodies. These antibodies remain free upon addition and are washed off during washing. Substrate is added, but there is no enzyme to act on it, so positive result shows no color change. Animated video overview of competitive ɨʻlaɪzə, multiple and ready-to-use ɨʻlaɪzə. A new technique uses a solid phase made up of an immunosorbent polystyrene rod with 8 to 12 protruding ogives. The entire device is immersed in a test tube containing the collected sample, and the following steps are carried out by dipping the ogives in microwls of standard microplates filled with reagents. The advantages of this technique are, the ogives can each be sensitized to a different reagent, allowing the simultaneous detection of different antibodies and/or different antigens for multiple target assays. The sample volume can be increased to improve the test sensitivity in clinical, food and environmental samples. One ogive is left unsensitized to measure the non-specific reactions of the sample. The use of laboratory supplies for dispensing sample aliquots, washing solution and reagents in microwls is not required, facilitating the development of ready-to-use lab kits and on-site testing. Applications. Because the ɨʻlaɪzə can be performed to evaluate either the presence of antigen or the presence of antibody in a sample, it is a useful tool for determining serum antibody concentrations. It has also found applications in the food industry in detecting potential food allergens, such as milk, peanuts, walnuts, almonds, and eggs. ɨʻlaɪzə can also be used in toxicology as a rapid presumptive screen for certain classes of drugs. The ɨʻlaɪzə was the first screening test widely used for HIV because of its high sensitivity. In an ɨʻlaɪzə, a person's serum is diluted 400 times and applied to a plate to which HIV antigens are attached. If antibodies to HIV are present in the serum, they may bind to these HIV antigens. The plate is then washed to remove all other components of the serum. A specially prepared secondary antibody an antibody that binds to other antibodies is then applied to the plate, followed by another wash. This secondary antibody is chemically linked in advance to an enzyme. Thus, the plate will contain enzyme in proportion to the amount of secondary antibody bound to the plate. A substrate for the enzyme is applied, and catalysis by the enzyme leads to a change in color or fluorescence. ɨʻlaɪzə results are reported as a number. The most controversial aspect of this test is determining the cutoff point between a positive and a negative result. A cutoff point may be determined by comparing it with a known standard. If an ɨʻlaɪzə test is used for drug screening at workplace, a cutoff concentration, 50 ng/ml, for example, is established, and a sample containing the standard concentration of analyte will be prepared. Unknowns that generate a stronger signal than the known sample are positive. Those that generate weaker signal are negative. Dr. Dennis E. Bidwell and Alistair Voller created the ɨʻlaɪzə test to detect various kinds of diseases, such as malaria, Chagas disease, and John's disease. ɨʻlaɪzə tests also are used as in vitro diagnostics in medical laboratories. The other uses of ɨʻlaɪzə include detection of Mycobacterium antibodies in tuberculosis, detection of rotavirus in feces, detection of hepatitis B markers in serum, detection of enterotoxin of E. coli in feces, detection of HIV antibodies in blood samples. See also, immunoscreening, lateral flow test, magnetic immunoassay, plaque reduction neutralization test, secretion assay, notes and references. External links.